So the meeting is getting recorded now. Am I audible?
Yes, sir.
Yes, sir.
Okay, great. So good evening, everyone. Welcome to today's lecture. I would just quickly go back to my slides. So, as you can see, today's lecture is based on forensic biology and wildlife forensics. Before we begin, I want you to close your eyes and imagine for a moment that you are in a crime scene. There is no eyewitness, no CCTV footage, no confession. The room is silent, but on the floor, a faint reddish brown stain. On the window sill, a single strand of hair. And on a glass, a smudge of saliva which is invisible to the naked eye. Okay? To an untrained person, these stains are just debris, right? They are useless to them. But to a forensic biologist, a trained professional, they are witnesses which are willing and waiting to speak. Is it clear till now?
Yes, sir.
So, this is where our journey into forensic biology begins. This is exactly why I named this lecture Silent Witnesses: Blood, Genes, and the Wild. Today, we will be discussing the basics of forensic biology, serology, and wildlife forensics. Standing before you today on this platform, I do not want you to see this subject merely as a laboratory discipline or an exam paper. I want you to understand it like a bridge, as a bridge between life sciences and justice. So forensic biology is yeah. So forensic biology is a bridge that is connecting both of these, right? It is not just about blood or DNA, it is about transforming biological traces into legally admissible truth. It is the science that interprets life at molecular level to reconstruct the events at the human level. In the last lecture, I already told you how crucial this factor is and how even a small mistake can make the evidence inadmissible in court. Okay, let me define it formally first. So, forensic biology, as you all may know, is the application of biological principles and laboratory techniques to the matters of law and criminal investigation. But definitions are dry, so let me rephrase it in a way that you will remember it. Whenever material derived from a living organism becomes evidence in a legal case, forensic biology takes charge. If it comes from a human body, an animal, a plant, or even microorganisms, and as long as it is relevant to a legal question, it falls within our domain. Now, let me ask you something. If I hand you a dried stain and ask you, is this blood? Can you answer immediately? You might say yes based on color, but what if it is rust? It can be rust, right? What if it is paint? It can be ink. There are so many possibilities. So, science does not work on assumptions, it works on validation. This is the first mindset shift that I want from you. Forensic biology begins where assumptions end. Am I audible? Clear till now?
Yes, sir.
Yes, sir.
Okay. So, historically, this field did not start with DNA. It started with simple observations. In early days of criminal investigations, experts relied on blood grouping system. Who can tell me what are the types of blood groups? Would anyone like to tell me?
Basically, there are four blood groups: A, B, AB, and O.
Okay, and there is also Bombay phenotype, right? Okay, so let's not go into too detail for now, but for you to understand, this ABO blood grouping system could narrow down the suspects but not identify them quickly, right? They could not uniquely identify If you remember, in the last class, we discussed class characteristics and individual characteristics, isn't it? So, imagine a city where millions shared the same blood group. That was the limitation which investigators faced. The science was helpful, but it was not decisive enough. Okay, sorry. So, everything changed in 1984 when Sir Alec Jeffreys discovered DNA fingerprinting. For the first time, biological material could be linked to a specific individual with extraordinary statistical certainty. It was revolutionary because cases that once depended on eyewitness testimony could now rely on molecular evidence. Innocent individuals were exonerated and hidden perpetrators were identified decades later. Biology at that time became the gold standard of forensic identification. Here, I want you to pause and think carefully. DNA did not make forensic biology powerful overnight, right? It amplified a foundation that already existed. Serology, microscopy, biological screening tests, and evidence handling protocols. Without proper collection and preservation, even the most advanced DNA technology is useless. The science begins at the crime scene, not in the laboratory. We discussed that already in our crime scene management lecture. Understood? Till now, everything is clear, right?
Yes,
Yes,
sir.
sir.
Okay. If you have any doubts or any questions, feel free to ask me. Okay. If you need me to repeat
Yes,
anything,
sir.
because today my voice is not at its best, so I can understand. If you are having any kind of issues understanding, then please ask me questions. Okay. So now let us step into the laboratory, but mentally, right? In a virtual meeting, we cannot physically access the laboratory, but in your mind, you can access it. So when a biological sample arrives, we do not immediately extract the DNA. First, we ask some fundamental questions. What are those questions? Is this sample even biological? That is the first question. Second question, is it human? What type of biological fluid is it? Is it blood? Is it semen? Is it saliva? Is it any vaginal secretion? Is it anything else? Each question requires specific understanding and testing strategies. Each has different evidentiary implications. And forensic biology is systematic. It moves from general to specific, from screening to confirmation, from indication to identification. Clear till now?
Yes, sir.
Okay. So one more thing, guys, I have not included too much in the PPT because you can see it's only 18 pages at most, I guess, 18 slides. I would be speaking. Okay. You guys should understand, and after the session, I would share you all the relevant sources, relevant materials, extra PPTs and notes, whatever I have on this topic, whatever I think is relevant for you guys, I will share it to you. Right? For now, these are just some visual representations so that you do not get bored listening to me, right? And you can also relate it. You have something as a reference. That's the point. But I'm not exactly going slide by slide. That's not a problem, right? Am I
No,
audible?
sir.
Okay, great. Fine. Now consider this. Forensic biology is not restricted to humans only, is it? It extends to forensic entomology, where insect developments help estimate time since death. It extends to forensic botany, where pollen grains can link a suspect to a location. Sorry. It includes wildlife forensics, where DNA identifies illegally trafficked animal products. It even includes microbial forensics in case of bioterrorism. When you understand this breadth, you realize that forensic biology is not just a narrow discipline. It's a vast biological toolkit that is serving justice. However, I must also be honest with you from the very beginning. Forensic biology is powerful, but it's not infallible, right? Contamination can compromise results. This is a fact. Improper handling can destroy evidence. Interpretation of mixed DNA profiles can be complex. Statistical misrepresentation in court can mislead judges and juries. Therefore, this subject demands not only technical competence but also ethical integrity. A forensic biologist must be scientifically rigorous and legally cautious at the same time. You will notice throughout the study of this topic that we constantly balance these three dimensions: science, law, and ethics. I have also tried to show it here, right? Science, law, and ethics. All these three need to be balanced. Sorry. So the science provides the data. Law defines admissibility and standards. Ethics governs responsibility. If one of these fails, then the justice suffers. That is why your training must be interdisciplinary. You are not just scientists, you are potential expert witnesses, educators, and custodians of truth. Let me also address something very important for those of you who are serious about research or advanced study in forensic biology. Modern forensic biology is rapidly evolving. Techniques like mitochondrial DNA analysis, YSTR profiling, and next generation sequencing are expanding our capabilities. We are entering an era where genetic data can sometimes predict ancestry or externally visible traits. Phenotype, you can say. This raises profound questions about privacy, consent, and state power, right? It's about ethics. As future professionals, you must not only master the techniques but also critically evaluate the implications that they are going to have on us as humans, as society. Okay? I want this specific lecture to function differently from a typical workshop or virtual lectures that we usually take. I expect you to question. That's something I have been repeating from day one. I expect you to challenge. If I say something, ask why. If a method works, ask me how. If a result is presented, ask about its statistical foundation. Forensic biology thrives on skepticism. Blind acceptance has no place in science, let alone forensic science. So, as we formally begin, remember this: every cell carries a story. Each biological trace is a fragment of a narrative. Our role is not just to dramatize it, but to decode it with precision. We translate molecular signals into reconstructive insight. We convert the biological remnants into courtroom testimony. Is it clear till now?
Yes, sir.
No difficulties understanding, right?
Yes, sir. No difficulties.
Okay, great. Sorry. Yeah. And perhaps the most important lesson that I want you guys to carry from this introduction is that forensic biology is not about catching criminals alone. It is equally about protecting the innocent. The same DNA that convicts can also exonerate. The same biological science that implicates can also liberate. The dual responsibility defines the moral gravity of this field. Now we will move more systematically from the nature of biological evidences to laboratory methodologies, from DNA extraction to statistical interpretation, from case studies to courtroom dynamics. But today I want you to leave with intellectual curiosity ignited after this lecture. Is that clear?
Yes, sir.
Yes,
Clear.
sir.
Great. Because once you begin to see biological evidence not just as stains, not just as hair strands or microscopic cells, but as enclosed truth, you will never look at the crime scene the same way ever again. Because DNA profiling revolutionized crime scene investigations. Criminal investigations, forensic laboratories relied heavily on serology, the study and identification of bodily fluids. Okay, sorry. And let me be very clear, even today, serology is not obsolete. It is the gateway to DNA analysis. If you cannot correctly identify the biological material, you cannot proceed intelligently to genetic testing. Okay. Let us slow down and understand what serology actually does. Do you guys have any idea what serology is? Have you studied it in any of your semesters?
Uh, Sir, is serology the study of body fluids? Maybe.
Yes. Yes, it is the study of body fluids. Anyone else? Sorry. Anyone else?
So basically, the study of like blood serum to detect antibodies.
Okay, it's not just blood serum, but also body fluids, right? Okay. So, serology basically answers some foundational questions. First, when you are getting a stain, is that stain blood? Second, is it human blood or not? Is it semen, saliva, or something else? What exactly is this biological material? And where is it located? Okay. Only after these are answered do we move forward. When investigators arrive at a cleaned or darkened crime scene, I have just given a reference in the left hand side. Okay, you can see the cursor here. But washing removes color. Okay, as the criminals attempt to wash it away. But not necessarily the chemical components like hemoglobin that is in blood. It contains iron. Luminol reacts with iron in hemoglobin in the presence of an oxidizing agent. This reaction produces chemiluminescence. Chemiluminescence means luminescence, the emission of light, which is due to chemicals. Okay, it would usually be seen as a blue glow, like here. So, notice something important here. This is not fluorescence, it is chemiluminescence. It is light produced from a chemical reaction without any external light excitation. Okay, imagine a pitch dark room. Investigators spray luminol. Suddenly, a blue sticks appear across the floor, forming patterns like arcs, drops, and smears. These are not just glowing patches, these are basically the bloodstain patterns. They will tell you the directionality, they will tell you about the movement, they will tell you whether blood was wiped, splashed, or cast off from any weapon. They will help you reconstruct the entire crime scene. Now, I will just give you one challenge. Tell me whether luminol is only specific to blood. What do you think? Tell me.
Sir, repeat again, please.
Yes, Rugved, I am asking whether luminol is specific to blood.
No,
Anyone?
sir.
Sorry.
It's not, sir.
Okay. So, any idea why is it not specific to blood? Sorry.
So it can give false positives as well. For household cleaning things, it's specific to hemoglobin and also can give false positives for bleach and certain chemicals.
Good. So, the answer is no, because it reacts with some other oxidizing substances too. Okay. Like bleach ho gaya, there are certain metals, there are plant peroxidizes, right? That is why luminol is called a presumptive test. You guys know the difference between presumptive and confirmatory tests, right? Yes.
Yes, sir.
Okay. It indicates that possible blood presence might be there, right? It indicates a possible blood presence, but it does not prove it conclusively. That is the difference. This distinction, presumptive versus confirmatory, it is fundamental in forensic biology. Okay, let me just open my slide back again. Let us now move to Kastle-Meyer test. This again is a presumptive screening test. Presumptive screening test. Yes. So this test basically uses phenolphthalein. You must have seen phenolphthalein in your labs, right? Some of you might have also performed the bribe trap. Have you?
No,
Yes,
sir.
sir.
Okay, someone has not, but it's totally fine. In near future, you would also be performing the bribe trap cases. It is nothing but in forensic chemistry. I would just tell you a brief in forensic chemistry. So this phenolphthalein is normally colorless. So in cases of corruption, when phenolphthalein is an indicator, okay, it can indicate in presence of base that is alkali, so it turns into pink color. So you can catch the corrupt people who are taking bribes, you can catch them red-handed or rather pink-handed in this case. Sorry. So phenolphthalein is usually colorless, but in the presence of hydrogen peroxide and peroxidase like activity or hemoglobin, it oxidizes and turns bright pink. A similar mechanism as I told you, right? In that case, it usually uses alkali, okay, in cases of bribe trap. This reaction is fast and almost dramatic. A cotton swab touches the suspected stain, add some reagent, add peroxide, and within seconds it would turn pink. But again, we must think critically. Why does it turn pink? Because hemoglobin catalyzes the breakdown of hydrogen peroxide. Sorry. Okay, just a second. Yes, am I audible? Am I audible?
Yes, sir.
Yes,
Yes,
sir.
sir.
I'm
Yes,
really
sir.
sorry, guys. Due to the cold, I'm having a little difficulty in speaking non-stop. Right. So where were we again? Peroxide, right? Yeah. So why does it turn pink? That's the fundamental question. Because hemoglobin catalyzes the breakdown of hydrogen peroxide. That peroxidase-like activity triggers oxidation. And again, is it specific only to blood? No. Some plant materials and certain chemicals can also produce false positives. Therefore, a positive Kastle-Meyer test tells us this is likely blood. It does not tell us that it is definitely human blood. This is where confirmatory testing comes in. One classical confirmatory test is the Takayama crystal test. In this test, under a microscope, specific hemochromogen crystals form if blood is present. This is more specific, but even does not answer whether the blood is human. To be clear, hemochromogen is a bright red compound that is formed by the combination of reduced heme, that is ferroheme, with a nitrogenous base such as denatured globin or pyridine. It is a derivative of hemoglobin and is created in forensic tests to identify bloodstains, often appears as a pink colored needle shaped, as you can see in this picture, crystals, which particularly when using the Takayama reagent. Clear till now?
Yes,
Yes,
sir.
sir.
Okay. To determine the species origin, whether the blood is human or animal, we use the precipitin test. This is immunological. Antibodies raised against human serum react specifically with human antigens. If precipitation occurs, it confirms human origin. Notice how the science progresses. Locate possible blood first using luminol. Second step is presumptive test. Kastle-Meyer. Confirm if blood is present using crystal test. Confirm the species origin, whether it is human blood or not, using precipitin, and only then we can move to DNA profiling. This layered approach protects the scientific integrity. यहाँ तक पूरा क्लियर है?
Yes, sir.
Sir.
Okay. Now let us move beyond blood. In sexual assault cases, I'm audible, right?
Yes, sir.
Yes,
Okay.
sir.
Yeah. In sexual assault cases, semen detection was historically critical. The acid phosphatate test is one example. Semen contains high levels of acid phosphatase enzyme. When a substrate is added, a color change indicates its presence. But acid phosphatase is again not exclusive to semen. It is just present in higher concentration there. So again, presumptive, right? For confirmation, microscopic identification of spermatozoa was traditionally used. Under the microscope, sperm cells have a distinct head and tail morphology. You can see in the left direction here. Okay. But here comes the complication. What if the assailant had undergone vasectomy? What if the sample is degraded? What if assault involved azoospermic semen? You know azoospermia, right?
Yes, sir.
Okay. Azoospermia basically is कि sperm cells won't be present. Again, modern forensic biology then relies on prostate specific antigen testing. Okay, that is PSA. And DNA extraction from epithelial cells. Now, let me tell you something that I emphasize strongly to my juniors and students. Serology is not just glamorous. It does not produce billion to one statistics like DNA profiling, but it is the screening filter that ensures we analyze the correct material. It reduces unnecessary DNA testing. It preserves laboratory resources. It prevents misinterpretation. There is also a forensic philosophy embedded here. Okay. Now, color changing reactions like luminol glowing blue, phenolphthalein turning pink, these are not magic tricks. They are biochemical reactions governed by enzyme kinetics and oxidation reduction principles. If you understand the chemistry, you control the interpretation. If you simply memorize the test, it means you can have errors, right? You are risking it for errors. Now, let me pose you a question. Suppose luminol glows strongly in a kitchen. What does it mean? Does it mean that a murder happened here? Or could it be anything else? What do you guys think? Sorry. Yes, anyone? Hmm.
So it doesn't rule it out, but it also doesn't confirm it.
Yes. Because it could be murder, it could also be bleach from cleaning, right? So the context always matters. Forensic biology operates within context. Laboratory findings must be interpreted alongside scene reconstruction, witness accounts, and investigative logic. Before DNA database existed, these serological techniques helped narrow down the suspects using blood grouping system like ABO typing and enzyme polymorphisms. Although not individualizing, they could exclude the suspects, and exclusion is also very powerful. Science often works by eliminating the impossibilities. So, as we conclude this section, I want you to appreciate this phase of forensic development. Serology, it basically represents the foundation, the disciplined, the methodical identification of biological material using biochemical reactions and immunological specificity. It taught the investigators and scientists how to question evidence before jumping to conclusions. It introduced the structured testing hierarchies. It laid the groundwork upon which DNA profiling was later built. Okay. Before we move ahead, let me ask you, if you had to defend the reliability of luminol in a court, how would you explain its limitations without weakening the prosecution's case? Any ideas? Think about that. Because this is where science meets law and your forensic mindset is tested. You guys understood my question, right? If you want, I can repeat it once again. I
So
said,
please repeat again.
yes. If you had to defend the reliability of luminol in court, how would you explain its limitations without weakening the prosecution's case? क्वेश्चन समझ आया? थोड़ा और elaborate करके बताता हूँ. You are the expert witness who is giving the expert witness testimony. Court has asked you, आपने रिपोर्ट में लिखा है कि लुमिनॉल से मुझे पता चला कि वहां पे ब्लड स्टेन्स था, राइट? बट अगेन वी डिस्कस दिस कि लुमिनॉल इज नॉट कंक्लूसिव, तो उसकी लिमिटेशंस है. बट अगर आप लिमिटेशंस बताओगे डायरेक्टली जज को, तो व्हाट वुड जज डू? जज वुड से कि अरे फिर तो केस ही नहीं बनता है, ब्लड ही नहीं था, देयर वाज नो ब्लड, लुमिनॉल कैन गिव फॉल्स पॉजिटिव्स. सो यू हैव टू प्रेजेंट योर केस इन सच ए मैनर दैट आपका जो केस है, वट एवर रिपोर्ट यू हैव रिटन, वो प्रोसिक्यूशन के तरफ से है, राइट? यू आर प्रोसिक्यूटिंग दैट एक्स वाई जी परपिट्रेटर कि हाँ उसने मर्डर किया है, दैट इज वाई देयर वाज सम ब्लड इन्वॉल्व्ड, which can be proved by the luminol. Got my question? So without weakening your own case, how will you defend yourself? How will you explain the limitations to the judge? Think about it, because this tests your forensic mindset, and I want you guys to think about the answer. You can later let me know. Each one of you have my number, so just text me whatever you think is the answer. Whatever, it can be a voice note, it can be a text, it does not need to be a report, but whatever you think the answer can be, just let me know. Got it?
Yes, sir.
Any doubts?
Yes, sir. Sir, I want to ask something.
Yes, sure.
Sir, just like blood, when we get into a luminol test,
Hmm.
it like it gives a chemiluminescence, right?
Yes, it gives
So in
chemiluminescence.
yes, so in case of bleach, like it also give chemiluminescence. So
Yes.
will it be get faint instantly, or the time period will be varying?
See, now the question is, even if you are getting chemiluminescence from other oxidizing agents, it can be bleach, it can be anything else. It does not necessarily need to be blood. How would you prove that it is blood? That is why I said कि we move in a structured manner. First step is to exclude. You would exclude some possibilities. When you see there is some stain, you know at least this is not other biological material, or maybe it is not paint, it is not ink. Then you slowly move towards confirmatory. First saw that presumptive test says that there is something. It does not necessarily need to be blood, but there is something. Now you need to identify that something. So you will first put it in class characteristics, then you will put it in its individual characteristics, and you will individualize it. How will you do that? As I explained, first you will go and you will start by presumptive test. First you have to identify there is something. Now if that something is blood, then your presumptive test, the second one that I explained, let me just go back to the slides so that I can explain better. Sorry. Here, first was luminol test, which says that there is something. Next, you go to Kastle test, where you know that if that something is blood. If yes, then you will go to the next step. If no, then it is something else, probably bleach or probably something else. Now, as an investigator, if it is bleach or if it is any other kind of oxidizing material, does that concern you? No, because you have to take care of a crime, you have to solve that, right? Whether they have cleaned their kitchen counter with bleach or not does not matter to you. To you, if it is blood, then it is important, otherwise it is not that important. If it is Castlemayer if tells you that yes it is blood, then it is likely blood, but not definitely human blood. Then you will need to test whether it is human blood or some chicken blood or something else. Then you move towards the certainty, the confirmatory test. Next, you go to Takayama test. It confirms that yes, it is blood. 100% it is blood. And then still you do not know what species does this blood belong to. Then you go for immunological reaction, that is precipitin test. What do? You first take some antibodies which are human specific. You take some antigens that are human specific, add them, antibody and antigen will combine and they will form some reaction, they will cause coagulation, right? And precipitation will form, like there will be some kind of precipitate formed, right? This will confirm that yes, this actually belongs to a human. Now is it clear?
Yes, sir.
Okay, great. Again, where were
So
we?
another slightly related question.
Sure.
So what if it is the example you've given us? What if we find luminol to be reactive on a kitchen counter, say,
Hmm.
but there has been a murder in that place and that could be blood, but they've tried to erase the evidence using bleach.
Hmm.
Would we get enough blood to perform the confirmatory tests?
Now, even if you are using bleach, you cannot directly remove all traces of it, right? Because that brings us to the very basic thing that we studied in the first lecture. Remember, Lucard's exchange principle. Whenever there is a crime happening, some traces will definitely be left. No
No, sir.
perpetrator
What I
can be smart enough to remove all traces. I got your question. Now, the point is whether or not would depend on multiple circumstances. Now, let's say there is a perpetrator who has OCD, who is extremely skeptical about it. Before the investigators arrive, he has washed that place with bleach at least 20 times. Do you think there is any possibility you will still find some blood traces? No.
no,
But what usually happens, I'm telling you, is if I commit a murder, first thing in my mind is not about how to remove the traces. First thing is panic. Oh my god. If I have committed a murder and if anyone else discovers it, it does not necessarily need to be an investigator. It can be any random person in my neighborhood. They will call the police and I will be inside the jail. Before I know it, first thing is panic. And whenever a person is panicking, would he be in the right state of mind to remove all traces? No. At that time, they will
So
miss it mostly.
somewhere the traces would be found.
Yes, somewhere the traces might be found. Now, that's what I was talking about. If I am extremely skeptical and I'm getting enough time, I'm getting, let's say, 7 days to erase the evidence, then you know, right, that the longer the time it takes to reach the crime scene for the investigators, of course, more the evidences will degrade, especially the biological evidences, and then the analysis will also be faulty, right? Sorry. If I, as the suspect, am getting enough time, then I will clean every nook and corner. But usually that's not the case. Whenever a murder happens or whenever something happens, usually the police detects it and they arrive at the crime scene. So before that, I will not get ample amount of time to clean it 20 times. Yes, I will try my best to clean it. But do you think एक wash में blood चला जाएगा? No. Some or the other traces will be left because not everything that we treat as evidence is visible to the naked eyes. It does not need to be visible to the naked eyes. It can be invisible. But chemical tests would determine the presence of it, right? Ha, quantity will be very less. You need to use extremely sophisticated instruments to detect them in trace quantities. That is one of the reasons why some biological evidences are also known as trace evidences, because they will be found in very trace quantities. Is that clear, Yogini? Vanshika, is it clear?
uh, yes, sir.
Yes, sir.
So, now coming to the question that you actually asked me, that what quantity do I need to assure, right? Like कितना bleach हम डालेंगे तो blood will absolutely disappear from it. That was the question, I guess. Am I understanding it correctly?
No, so the question was just that if it's been cleaned once or twice, would we get enough to perform confirmatory tests?
Yes. So that's what I was talking about. Uh, कि एक दो बार भी करेंगे तो yes there are chances. Now yes, I won't say there are like 99% chances, because if there is an undisturbed crime scene you will 100% get it, but उसको इरेज करने का अगर थोड़ा सा भी ट्राई किया गया है तो डिपेंड्स हाउ एफिशिएंटली दे हैव ट्राइड टू क्लीन इट. ठीक है, इफ दे हैव नॉट डन अ वेरी गुड जॉब देन देयर आर वेरी हाई चांसेस यू वुड फाइंड द एविडेंस. ठीक है, एग्जैक्टली कितना या कितने टाइम क्लीन करना है वो तो देखो लेबोरेटरी में तुमको एक्सपेरिमेंट करके ही पता चलेगा कि हाउ मेनी टाइम्स डू यू नीड टू क्लीन इट. गॉट माय पॉइंट?
Yes, sir.
Okay, chalo. Now let's move on to the next topic. I will quickly go back to my slides. So I hope the question that I have asked is clear, right? So you can just let me know the answers through WhatsApp. Take your time, think about it. Since we are already running out of time, I will keep the rest of the sections very brief. Let's talk about the DNA revolution first. The most profound advancement in the field occurred in 1985 when Sir Alex Jeffreys, as I have previously mentioned, discovered, or rather developed, modern DNA profiling. Moving beyond basic blood typing, we could now use polymerase chain reaction, that is PCR, as a molecular photocopier to reliably replicate tiny degraded DNA traces by evaluating STR, short tandem repeats. Right? We can create a highly individualized genetic barcode to distinguish one suspect from another. Again, this brings us back to Yogini's question. Even if we found a very slight trace amount, then we can perform PCR on that polymerase chain reaction. We can replicate that small amount of DNA that we are finding, and we can further analyze them under very sophisticated instruments, and we would get a good result, right? Again, let me share a very famous narrative about Linda Mann and Dawn Ashworth. Sorry. So these are the two girls that tragically were murdered in the UK during the 1980s. This is a case study. I don't think I have included it in the slides. Have I? Okay, okay, I have. Okay. So you can see the spellings here. Linda Mann and Dawn Ashworth. UK 1980 का case है. So, investigators basically found identical biological evidence at both scenes, leading them to strongly suspect a local teenager who was named Richard Buckland. However, the historic scientific first DNA profiling proved that Buckland's genetic material did not match the crime scene evidence. That's why, while previously explaining serology, I repeatedly emphasized on exclusion, because it does not only convict, it can also exclude some people from suspicion. Okay. Now, next, what I'm going to discuss is often called the first manhunt. To find the real killer, the police conducted the world's first DNA based manhunt by testing 5,000 local men. The actual murderer, who was Colin Pitchfork, initially evaded capture by persuading a friend to submit a blood sample on his behalf. Once the falsehood was discovered, Pitchfork's true DNA was tested and yielding a perfect match for the sample that was found in crime scene. This led to his imprisonment in 1988. After the class ends, I would share the case study with you guys. And I want you to analyze the turn of events in this case and write it down. You can also include a modern case study to show the differences in both these cases. One is primitive and one is modern, right? So, what are the types of technologies used, what processes were used, and how exactly did it happen, right? You can show the contrast. Is that clear? That would be your task for this weekend. Sorry. Any doubts?
No, sir.
Okay, I would share the entire case study with you guys, and the case study already has everything in it that's required. However, you guys are required to share your analysis of it, and you can find one case study that is modern, and you can compare and contrast both of them. Moving on. Sometimes the standard DNA testing fails. Sorry. So sometimes the standard DNA testing falls short when crime scene evidence is heavily degraded or features multiple contributors. In cases which involve multiple males, forensic biologists usually use Y chromosome analysis to isolate and target only the male DNA fraction. For ancient bones or rootless hair shaft, we usually turn to mtDNA, that is the mitochondrial DNA, because it survives much longer than nuclear DNA in that environment. Beyond bodily fluids, we also study microscopic plant pollen and the spores to solve crimes. This is known as forensic palynology. Okay, sorry. It is fascinating specialty known as forensic palynology. And I said fascinating because plants disperse pollen in highly unique geographical patterns. A single microscopic grain can act as a fingerprint for a specific location. These microscopic palanomorph are incredibly doable, okay, and frequently become trapped in the suspect's clothing, hair, or muddy shoes. Let's talk about the Danube River murder mystery. This is basically a famous 1959 case in Austria where a man mysteriously disappearing during a trip down the Danube River. Police examined mud from the suspect's boots and discovered a 20 million year old fossilized pollen grain from the ancient Miocene age. Because of this specific fossil only existed in localized forest region, they confronted the suspect and he quickly confessed and led police to the grave. Now, I have a very important question to you guys. Do you think, like, what do you think is the most crucial part of this particular case? What was the turning point in this case? What do you think?
So the finding of the pollen grain.
Okay, let me quickly come to the screen. Yes. So, who answered this question?
So Vanshika.
Vanshika. Great. And what about others? What do you guys think? Do you guys also think that it was the turning of, the finding of the pollen grain that changed this case?
So the finding of the pollen grain in that specific region, I mean, belonging to that specific region.
Okay. So Yogini has similar answer. Someone else? Disha? Rugved? Krishna? Avani? Kanika? Anyone else?
So maybe the soil and the pollen grain, because the soil of that particular region may be very like from
Okay.
graveyard.
So, guys, I know we are studying forensic palynology. I know it's about pollens, but please think a little out of the box. What is the most important turn of the event here? Analyze it.
The, the hard remains maybe of the body that in that graveyard, the grave box.
Okay. So now, see, I agree that the remains of the person is very crucial, but how did the police reach there? Think.
So like by finding the mud on the boots, they could like track the geographical location of the mud.
Yes, but where is the crucial part? The crucial part is not actually the circumstantial evidence that they found. Guys, it was not a primary evidence, it was just a circumstantial evidence. It could have been for any other reason, right? आप किसी भी रास्ते से चल के जा रहे हो, would you not get pollen stuck in your muddy boots? You would, right? Here, the most crucial part is actually the psychology of the perpetrator. As I mentioned, whenever I am committing a murder, the first thing is I am nervous, I am anxious, I am panicking. He collapsed. That murderer collapsed in front of the investigators. He broke down and he confessed. So the most crucial part here is actually the confession. Otherwise, तुमको pollen मिल जाए, circumstantial evidence मिल जाए, you would never be able to find the body. Because the pollen was not directly related to the body, it was related to a forest, a local soil region where they found it, right? There is an entire forest. How would you find in what exact location he had buried the body? Unless the perpetrator was so nervous that he himself confessed it. So this is actually the turn of event that I was talking about. I agree that pollens were important in this case, but single pollen could not prove anything. It is nothing but an circumstantial evidence. It was the psychological warfare. It was the perpetrator who was psychologically weak and confessed too easily, making this case very easy for the police to solve. Again, let's come come back to the slides now. I bet none of you expected that answer, but never mind. Okay, let's move on. So, forensic biology is actually a massive umbrella that covers several highly specialized fields of study. Depending on the evidence, an investigation might rely on sub branches like forensic anthropology to examine bones, which we will be discussing in near future in the next sessions. And in the upcoming sessions, it would be taken by Miss Ala El Sheikh, a Lebanese forensic scientist who would be discussing forensic anthropology and forensic odontology. Cool. Next, forensic entomology, which studies insects on a body, or forensic botany, which analyzes the plant matter. It also includes forensic serology, which we previously discussed, which focuses exclusively on the identification of bodily fluids before advanced DNA sequencing, definitely. Now, let's talk about the bug detectives, that is forensic entomology. Anyone would like to tell me about what forensic entomology is? Anyone has any idea? Any ideas
So
about
may I try?
entomology?
So basically, the study of the insect's life cycle in a particular human body or remains after the death of a human. So basically, the entomology deals with that, and it plays a crucial role in our forensic also because it can give us a time of death. So by studying the life cycle of maggots or bugs, we can like connect or link the time of death.
Great, excellent. So, forensic entomology is a fascinating study of insects, right? Such as blowflies, then beetles, right? And these help investigators determine crucial details about a crime scene. Like you mentioned, by analyzing the species of laid eggs on a cadaver means a dead body, and tracking their specific developmental stages, forensic entomologists can actually calculate the post mortem interval, which means when that person died, which tells the police exactly how long the victim has been dead. Are there no other options? Yes, there are. In forensic pathology, in forensic medicine, we can look at the rigor mortis, the liver mortis, and many other parameters are there. But in forensic biology, forensic entomology, insects also play a very major role in telling us this information. Got it? Again, slides. Right, we will talk about the case study now. So, understanding the precise life cycle of an insect from a tiny egg through its larval stages to a fully grown fly, it acts as a biological clock for the investigators. I hope you understood, right? And here you can see I have just given one small life cycle here. Okay. To see this in action, let's look at the historic Dr. Buck Ruxton case. This marked a major turning point for the field. When dismembered remains were discovered in Scottish ravine, investigators analyzed the specific age of the blowfly maggots infesting the bodies. The developmental timeline of these tiny flies perfectly matched the exact date Ruxton's wife and maid had gone missing. Sorry. This provided the critical evidence that was needed to secure his conviction. Fine. Any doubts? Any doubts?
So
No,
please
sir.
explain this once again.
Okay, the case study, right?
Yes, sir.
Okay. So, in this case, dismembered body parts were found. Dismembered बोले तो अलग अलग पार्ट्स को काट करके, लेट्स से लेग्स अलग, हाथ अलग, सब अलग अलग. Got it? So, the
Yes, sir.
dismembered, yes. Uh, any doubts?
No, sir.
Okay. Yeah, I was saying that dismembered remains were discovered. कहाँ पे? In Scottish ravine. Okay. And the investigators analyzed the specific age of blowfly maggots. ठीक है, blowfly के जो maggots infest कर रहे थे bodies में, उसको एनालाइज किया गया. नाउ सी हाउ इज दिस थिंग डन, कैसे होता है. लेट मी टेल यू इन वेरी ब्रीफ, बिकॉज सी टाइम हमारे पास बहुत शॉर्ट है. एंड यू गाइस आल्सो नो 1 आवर में पूरा फॉरेंसिक बायोलॉजी, एंटोमोलॉजी, पैरानोलॉजी, वाइल्ड लाइफ फॉरेंसिक्स, डीएनए, सेरोलॉजी सब कुछ कवर करना इज इम्पॉसिबल. नॉट जस्ट मी, आप किसी को भी बुला लो, दे कैन नॉट डू दैट इन 1 आवर और 1.5 आवर्स. डेफिनेटली नॉट पॉसिबल. बट आई एम ट्राइंग आप लोगों को एक बेसिक आइडिया हो जाए. For those who have already studied it in their semesters, आप लोगों का एक brush up हो जाए, right? You would also revise this topic. चलो, just एक brief idea देने के लिए, there is not just one species of an adult fly that is infesting that particular cadaver or dead body. There are multiple flies, and there is an insect succession. मतलब देयर कैन बी वन फ्लाई, ब्लो फ्लाई आया, फिर फ्रूट फ्लाई आएगा, फिर कोई और फ्लाई आएगा, एंड देन लाइक दैट देयर विल बी मल्टीपल 8 to 9 types of flies depending on that location and other things. They will be infesting that. सबका अपना लाइफ साइकिल होगा. एक फ्लाई आएगा, they will give egg, then there will be larva, pupa, adult fly, these things will go on, and then some other fly will come which will eat that, and they will give their egg. इस तरह से चलता रहेगा. जब फ्लाइज खत्म हो जाएगा तो बीटल्स आना शुरू होगा, बिकॉज फ्लाइज यूजुअली ईट द सॉफ्ट पार्ट. फिर जो हार्ड पार्ट्स जो बच जाएगा, दैट द बीटल्स विल ईट. सो सम बीटल्स विल कम, बीटल्स विल आल्सो ईट द फ्लाइज का जो भी लार्वा वगैरह, वट एवर दे विल फाइंड एग्स एंड ऑल, एंड देन वट एवर हार्ड पार्ट इज रिमेनिंग वो भी बीटल खाएगा. ठीक है. सो देयर इज एन इंटायर इंसेक्ट सक्सेशन. सो बेस्ड ऑन व्हाट काइंड ऑफ इंसेक्ट यू फाउंड and what kind of stages they are in. Like let's say I'm finding a blowfly, and blowfly ka I can find an adult, I can find egg, I can find its larva. So depending on what exactly you are finding, you can tell it like ki when was this person killed. So it acts as a biological clock, as I mentioned. ठीक है. So by analyzing the specific age of the blowfly maggot that they found, which were infesting the bodies, the developmental timeline of these tiny flies perfectly matched the exact date Ruxton's wife and maid had gone missing. So, whenever someone goes missing, what do? Even if you are the suspect yourself, तुम क्या करोगे? सबसे पहले you will go and file a police case report. Otherwise, the first question will come: if your wife is missing, why did you not report it? It means you knew where she went. It means you killed her, right? First thing that a perpetrator does is go and file a report and pose as innocent. तो पुलिस ऑलरेडी हैज अ रिकॉर्ड कि वो कब मिसिंग गई. एंड देन व्हेन दे आर गेटिंग द डिसमेंबर्ड पार्ट्स and estimating the post-mortem interval, they can see that okay, if on xyz date this person went missing, then on xyz date this cadaver was killed. This way they traced it, and critical evidence was provided for his conviction. So Dr. Buck Ruxton was sorry, was later convicted for the murder of his wife and maid. Clear? Hmm. Hmm.
Yes, sir.
Any doubts?
No, sir. Clear.
ओके, आप लोगों को समझ तो आ रहा है ना? बिकॉज आई फील मेरा गला वगैरह बहुत भारी हो गया है. आई डोंट इवन नो वेदर आई एम एबल टू स्पीक प्रॉपरली. आप लोगों को समझ आ रहा है ना?
हाँ सर,
ओके,
समझ आ रहा है.
ग्रेट.
Yes, sir.
So now let's talk about wildlife forensics, right? We will now be shifting away our focus from the human victims because forensic biology also fights environmental crimes through highly specialized branch which is called wildlife forensics. This field utilizes DNA fingerprinting and biological trace evidence analysis to identify illegally traded animal products such as poached ivory. You guys know what ivory is? You can also see an image for reference. You know what ivory is, right?
Yes, sir.
Okay,
Yes, sir.
okay. So, poached ivory is there, then there are exotic fur and protected horn, like one horned rhinoceros, है ना उसका एक वो रहेगा, and then there is a, if there is a white snow white fox something called like that, उसका white color का exotic fur है ना, and then there are many things for which uh, illegally the animals are poached on a daily basis. By tracing the exact geographic origins of these seized animal goods, scientists can help law enforcement dismantle international smuggling rings. Now imagine if you find a roadside seller selling ivory. How do you know whether it is authentic or not? You can trace it using the DNA profiling and all, right? And the other advanced techniques in this field. And once you know whether this is authentic or not, you can trace the geographical origin. Let's say it was African. So if it is African, what is it doing in India in a roadside black market? It means it was illegally smuggled, right? So the goods will be seized, the people related will be captured, and they will be punished according to the law, right? So, wildlife forensics essentially serves as a vital bridge between criminal justice and global environmental conservation. Whether they are analyzing a drop of animal blood left at the poaching camp or investigating the cause of a sudden mass die off in a local forest, these scientists ensure that endangered species are protected. Ultimately, their rigorous laboratory work enforces international treaties and holds wildlife traffickers legally accountable for destroying our planet's biodiversity. Now, that's all for today, I guess. So, as we begin to close today's session, I want you to pause and look at what we have actually touched till now. We started with serology, the color changing reactions that reveal hidden blood. We discussed luminol, illuminating washed away crime scenes. We explored the Castle-Meyer test turning pink within seconds. We moved into confirmatory testing, the species identification, right? And the disciplined hierarchy that prevents scientific overreach. We briefly stepped into entomology, where insects' life cycles quietly measure time since death. And we mentioned wildlife forensics, where DNA protects endangered species and combats illegal trafficking. Now, let me say something very honestly. What we have covered today is only the surface. Forensic biology is not a chapter, it is an entire ecosystem. Each domain we mentioned today, it can be forensic serology, DNA profiling, entomology, wildlife forensics, forensic palaeontology, or anything else, forensic botany. It can itself be taught as a full semester course. I could speak for weeks on bloodstain pattern interpretation alone. We could spend months discussing the DNA statistics and classroom cross contamination, courtroom cross contaminations. Forensic entomology requires deep understanding of insect developmental biology, temperature dependent growth rate, and ecological succession. Wildlife forensic intersects with conservation genetics, international law, and biodiversity science. That is precisely the point. You cannot master forensic biology in a single lecture, but you can develop the mindset that is required to pursue it. This field demands patience, it demands intellectual humility, it demands that you accept uncertainty while striving for precision. It demands that you understand both power and limits of your tools. Let me leave you with an image. Imagine standing in a laboratory at midnight. On your bench lies a single sample, tiny, nearly invisible trace evidence that was collected from a crime scene. That tiny fragment may determine whether someone spends the rest of their life in prison or whether an innocent person walks free. That is the weight on forensic biology. This science is not dramatic because it glows blue in dark. It is dramatic because it carries consequences. As future forensic professionals, researchers, or educators, your responsibility will not be to impress with terminology. It will be to uphold accuracy under pressure, to resist confirmation bias, to speak cautiously in the courtroom, to explain statistics clearly, and to admit limitations when they exist. That's why I gave you the task that was related to the limitation, right? Forensic biology in itself is very powerful, but only when it is practiced with discipline. And I hope today you have realized something deeper, that this subject is alive. It's evolving with technology. It challenges the legal systems. It raises ethical debates. It protects wildlife. It reconstructs the timelines through insects. It translates the microscopic traces into human narratives. If today's lecture has left you slightly overwhelmed, that's good. It means that you are beginning to see the scale of this field. In the coming sessions, we will narrow our focus, we will go deeper and technically, critically, and analytically. But for now, I want you to walk out of this virtual classroom with curiosity sharpened. Because the true beginning of forensic biology is not in the laboratory, it is in the questions that you ask when you see a trace. What story does this carry, and how do I prove it scientifically? That question will define your journey in this discipline. And if you stay intellectually honest, disciplined, and relentless in learning, you will not just study forensic biology, you will practice it with responsibility. That's all for today's session, guys. Thank you so much for being such a wonderful audience. If you have any questions, please feel free to ask me. So I'll be stopping my screen now. Sorry. If you have any questions, please feel free to ask me. No questions? Guys, no question can mean two things. Either I'm a good teacher, you have understood everything, or I'm a bad teacher and you are not willing to speak before me. You don't even want to ask questions. So, what is it? Did you guys understand or did you not understand anything at all? I will quickly check the chat sections where to find the recording. So, Kanika, I'll be uploading it. Don't worry. I'll be uploading it on YouTube and I will give it to you.